Someone turn the lights off. All right, so what I gave you before break, and I told you if you want to get ahead, you can get extra credit, was DNA and RNA. And at this point, if you didn't turn in, you didn't turn in. It was extra credit. This will be due when we're done covering it. We're doing most of it in class. Anything we don't do in class obviously won't be counted for a grade per se, but they'll be given another opportunity for extra credit. Just like the last study guide. Is that correct? All right. We've already gone over page 88, which is the first page. It has a gene that has a bacteriophage. We did it on the board before break, and we went over it. And we talked about this word genome and what it means. It's not up there? Hold on a second. Okay, now that you can see what's on the board. Genome is the word, and I said you need to know that that is the entire, all the DNA sequence, the DNA sequence for an organism. Is the genome for that organism. The example genome that they give you, you see that there's a lot of letters there. Do you agree that there's a lot of letters there? Are you all looking at this picture? Yeah. A lot of letters, and in the future I'll have this scanned in. I didn't bring it in yet. I didn't incorporate it into the video yet. What you see is the genome is the entire DNA sequence, and there's a lot of letters there, but it's a small number of letters compared to yours. You have about 3 billion, billion with a B, nucleotides. You have 23 pairs of these things called chromosomes. That's what you have. So you have 23 pairs. So you have a total of what? 46 is correct. 46 chromosomes. You have 23 from mom, and you have 23 from your dad. 23 of these. They have the same gene, but different sequences. And we're going to talk about what that means. What does it mean to have a gene? What does it mean to have a sequence? Look at those four. We went over this first page together before break. I don't think you should have any trouble converting that. I'm going to let you finish answering those questions. If you have trouble, you need to speak up tomorrow in class. I don't see an issue though. When we talk about prokaryotes, and we'll talk about these chromosomes in humans in a second, we talk about prokaryotes, we also talked about the second page. We say prokaryotes have this one circular DNA that we call a chromosome. They basically have one chromosome. What are prokaryotes? That's right, good. They're bacteria, or archaebacteria, right? There's two kinds of prokaryotes. And they are either bacteria or archaebacteria. Archaebacteria. These are two kinds of prokaryotes. What are prokaryotes? What does it mean to be a prokaryote? That's right. No nucleus or membrane-bound organelles. So you have, but they do have DNA. And they have these little things called plasmids. And all plasmids are little sections of DNA, smaller, you can think of them as small chromosomes. They have genes on there as well. They can have many of these, and they can share these with each other. So if you have a gene here that resists an antibiotic resistance, one bacteria can share that resistance with another bacteria, and you can spread antibiotic resistance in the population or amongst even different strains of bacteria. So you can have E. coli that's resistant to an antibacteria, and then that can make salmonella resistant to an antibiotic, which can then make Staphylococcus aureus, which is a staph infection, can become then antibiotic resistance, and you can get pneumonia to become antibiotic resistant. So these bacteria can share this resistance to antibiotics. Yeah.
It's called conjugation.
Conjugation is correct. Somebody's done their reading. That's right. So he's done his reading. By the way, I sent you a chapter that covers this as well, correct? Everybody has a chapter, an ebook on this unit, as well as this study guide. If you've done some reading, what happens is one bacteria can connect to another bacteria through something called conjugation. They make a little tunnel between them, and they send the little plasmids one way or the other, right? So they can send these little DNA sequences to each other to help each other out. They don't have sex. So in biology, sex is the mixing up of DNA. That's all sex is. Different animals, different plants, different organisms do it different ways. Plants have sex by spreading their pollen. They use bees and birds to help them get the DNA mixed. Humans mix up our DNA through having physical contact, penetration between the male and the female. Birds don't penetrate at all. They just touch. Excuse me. Fish fish lay the eggs in water, and then the male just sprays the sperm on top of them. And this sperm either gets to the eggs or not. So there are many, many different ways that sex occurs in different organisms. Fungi use things called spores. But in all cases, sex in biology is not about some kind of weird thing that you might see in a movie. It is about mixing up DNA. It's very, very, very important for the survival of a species to have what's called genetic diversity, having different genes in different populations. It allows a population to survive catastrophe. And we'll talk more about that as we get more and more deeper and deeper into this idea of evolution. Whether you agree with evolution on a grand scale or not, you have to understand the process. So conjugation is when DNA gets sent from one bacteria to the other, but there's no mixing up of the DNA, even though this chromosome here is its own, and this chromosome stays its own. Are we clear on this? All right, these do have ribosomes because ribosomes don't have membranes, but all the other organelles that we've discussed already that you should know already are not, these bacteria don't have them because they're prokaryotes. All right, let's move on. So when we talk about this idea of bacteria, in general, remember that prokaryotes have this circular DNA, so it can be very large or small, but they're circular, so there's no end, right? There's no 5' end or 3' end like we have with human DNA. Human DNA, eukaryotic DNA is generally linear. So you have a 5', a 3', a 5', 5', and a 3'. And they're anti-parallel. We've talked about this, right? Everybody should know this. This is something that's not new to you. But in both cases, on the DNA, somewhere along the DNA, there are these little sections. And where are they? Well, it all depends on the organism, on the chromosome. There's these little sections of DNA that do something. What do they do? What is this DNA? What does DNA do? What's the function of DNA? Remember we talked about macromolecules. We know proteins act as enzymes. They act as structural components. We know lipids can be structural in the case of phospholipids. They can store energy for long term. We know the function of the macromolecules. We know DNA's function is to do what? Hold information is correct. DNA's function is to hold information. But how do you get information? Information to do what? What do they do? What do information do? What to do? What? What does DNA hold? What kind of information does it hold? All right, genetic information. What does that mean, genetic information? When you have genetic information, what does that mean to you? Anything. Give me something. Yeah. Sounds like some people have to read, right? So let's start reading. Yeah.
Like the way you look. The way
Okay, it determines outcome. Genetic information determines the way you look is correct. Can I say structure? Is that okay with everyone? So the way you look is the structure of your face, the structure of your hair, the structure of your skin, the structure of your eyes, right? So structure of the organism. What else?
Would you consider like deformity as mutation?
Let's not talk, it's a good question. Let's not talk about mutation just yet. Let's talk about, first let's figure out what a gene is, right? And then we can talk about a mutation. And a deformity, by the way, doesn't have to come from a genetic mutation. It can come from being exposed to a chemical, like cigarette smoke can give you a deformity and not impact your genes. So that's another story. Yeah. Immunity. So I think I understand what you're saying, like where you're talking about immunity, like with the bacteria becoming antibiotic resistant. So you're right that it can affect, can I say function? How something functions, right? So in the case of immunity, with the antibiotic resistance, the genetic information that's been passed on changes the structure of the cell wall, which makes it resistant to the antibiotic. So you're actually changing a structure, but you can change function as well. How would you change that? How do you change that? Now that brings us to another question. You see how this all feeds into itself? It all feeds into itself. The next question is, how is it that you can change the structure and the function of an organism by changing the DNA? What is it you're changing, essentially? You're changing genetic sequence, which is what you mentioned with mutation. So you have a genetic sequence, A, T, G, C, T, T. How do you go from there to changing someone's face or hair color or making it a bacteria antibiotic resistant or making someone tall or short, thin or fat or fast or slow or smart or not smart? How do you go from the DNA sequence, which is here, right? A, T, G, C, whatever, to the outcome? Can I use the word, I'm going to use this word that everybody here has said over and over again. What is this word? Phenotype. How do you go from genetic information, another way for saying that is what? Genotype to phenotype. How does the sequence of DNA determine what something looks like? Yeah, but I'd rather you wait, but go ahead. Whatever you got to do. Can someone answer that? How? Anybody do the reading? Anybody? I feel like Ferris Bueller's teacher. Anybody? Anybody? How do we go from, so we're going to try to answer that. Let's answer it since you haven't covered it. I hope you guys do some reading now that you're back from break. So how do we go from genotype to phenotype? Well, what it is we have the sequence of DNA goes through one step, and then you end up with a protein. And that's it. That protein is going to do something. It could be a hormone that helps sugar get out of the blood. It could be a hormone that makes you grow tall. It could be an enzyme that helps you make a pigment in your skin, it makes you darker. Or a pigment, it could be an enzyme that doesn't work as well as somebody else's pigment, so you're lighter, right? So skin color. If you have one kind of enzyme, you make blue feathers. If you make another kind of enzyme, you have red feathers. Do you see that? So a protein in the form of enzymes, in the form of structures, maybe the protein that makes your hair has a lot of cysteines, or your hair is really curly. Maybe your hair doesn't have a lot of cysteines, so your hair is not curly, it's straight. So what you look like, your phenotype, is determined by what proteins you have. But then what proteins you have, what structures you have, is determined by the DNA. The DNA determines it. Is that clear? That should be like number one thing you put on an index card and kind of memorize. You need to know that the sequence of DNA eventually leads to a protein that does something. The protein can be an enzyme, the protein can be a structure, it can be bone, it can be a hormone, it can be all kinds of different proteins. And we went over the different functions of proteins. You should know that by now. So you should know that if you change a protein, you change the structure. You change what the thing looks like. You change the phenotype. All right, because phenotype is what you look like, what it looks like. All right, so how do we do that? Well, we go from DNA first, we make something called mRNA, and then we make protein. So going from DNA to mRNA, and we went over it a little bit for the last test, we call this transcription. This is transcription. So when we take DNA and we make mRNA, we call that transcription. When we take that mRNA and we make a protein, that's another process that's called what? Translation is correct. That's translation. Now it's important you take notes on this and you think about it. So when you go back and you read the chapter tonight, or you're getting ready for your activities, you have several activities we're going to do in class. They're all here in this workbook that you have. We're going to cut out the chromosomes. We're going to put them where they go. We're going to look at transcription and translation. We're going to actually look out. We're going to cut these things out and put them where they go. So make sure you're ready to do that during the week, this week. How would you prepare for that? 1, do your reading. Read, try to get ahead of the game. Read this. You know, ideally, you've already done some reading, but obviously sounds like some of you haven't. Secondly, you have a chapter in the textbook. You can read that. Thirdly, you can pay attention in class and get all you need to get from this class. Well, however you want to handle it. So 2 steps, transcription and then translation. You have to know these 2 steps. All right. So let's take a look at one essential reason why you need to do transcription and translation. There's a lot of different reasons, biochemical reasons, but there's a really fundamental issue with eukaryotes. Where is the DNA in eukaryotes? In the nucleus, correct. So DNA in a eukaryote is in a nucleus. It's here in the center. The ribosomes are where in a cell? Those of you that are outside the nucleus, where? On the ER. They're on the endoplasmic reticulum, the rough endoplasmic reticulum. That's why the rough endoplasmic reticulum is rough. And then you have the smooth area, right? So this DNA is inside a membrane. Let's be clear, and this is going to be tested. I'm going to test you on this. DNA does not leave the nucleus. That is non-negotiable. DNA cannot get through this nucleus. If we took a look at it, if we looked at it, if we kind of zoomed in on it, what you would see is there would be a membrane that would have a bunch of holes in it. These holes lead into endoplasmic reticulum, among other things. And so if we looked at the nucleus, it would look something like this. And it'd be, these things are called nuclear pores. That's not hard, is it? Pores mean holes. They're nuclear pores. They're holes in the membrane. Those holes in that membrane are to let something out, but it's not DNA. mRNA, correct. Excellent. See, you're so smart. God. I know, but you still look around you. Who's the only one that's answering? Think about it. Change your attitude. You can, oh my God, you could be so great. So, nuclear pores. The message has to get from inside the nucleus, right? From inside the nucleus to out here where the ribosomes are. But the DNA can't leave. So you have to change it to mRNA. Is that clear? All right. Now we saw a video, did we not? Before you left in December, we saw a video of that transcription happening, the little RNA polymerase clicking on. It went really fast. I even sent you the link, right? So here you have that DNA, you're going to make mRNA. And so transcriptions really, it should be really easy for you. Because all of you were able to do it in December. Hopefully you're still able to do it. So if you have an A, T, G, C, A, T, etc., what is going to be your mRNA? And by the way, the enzyme that makes it, the protein that makes this, is called what? What's the enzyme that does this? It's called, that's right, RNA polymerase. I don't care that you pronounce it any way you want, as long as you know what you can identify it, right? RNA polymerase is the thing that makes a polymer of RNA from DNA. It's just that simple. So if you have an A, what's the RNA polymerase going to make? U, because there's no T in RNA. Then A, then C, then G, then U, then A. So there's your RNA. This is what it's going to make. It's still the same. It's still the same. It's a message. It's just in a different form. That's all. This is easy, correct? Yeah. This should be easy. So all should be pretty straightforward. Now, there are some changes because it makes it RNA. So if you have the first thing it does, let's first take a look at what that DNA looks like. So you have this DNA. It's really long. It's a chromosome. We call this DNA a chromosome. That's what a chromosome is. Is that clear? It's a big, long DNA molecule. Actually, and each molecule is actually two molecules, right? They're held together by hydrogen bonds. So you got this giant molecule of DNA, two strands, from a 5' to 3'. This is your chromosome. It could be chromosome number 1. It could be chromosome number 2. It could be a lot of different things, right? You have 23 of them, 23 pairs. If you're a guy, you have 24 different kinds because you have an X and a Y, right? Even though you only have one of each, you still have 24 different kinds, right? Because you have an X and Y. Men have genes that women don't have, right? Yeah. Yes. So with the chromosome, the entire chromosome is not the message. What is the message called? The things that make the protein, things that the RNA polymerase is going to change, is going to make it to RNA. What do we call that? A gene, correct. So that thing, and if you remember from the video, if you remember from the discussions we had, there are numerous genes on a chromosome, many different genes on a chromosome, but they are different. So this gene here, maybe this talks about height. Maybe this makes an enzyme that impacts height. Maybe this gene makes insulin. So this is going to impact how well you're able to regulate sugar. Maybe this gene here is helps deal with skin color.
Melanin.
It might have to do with melanin. Maybe this one here has to do with how many impacts how many toes you have. So if you have, so you might have an extra one or whatever. Yep. So a lot happens a lot. Fingers or toes?
But ain't had no bone. It had a bone. I always had it like teeth my whole life.
That's different. Actually, you can get it removed anyways, but it's interesting. Yeah, there's a lot of different. Listen, and everybody has a different sequence. I'm really glad you shared that because you're the first kid that said I knew.
You couldn't move it?
Statistically, I can tell you that it's rare, but statistically it's unlikely that in a school you would like this as big as this, so you wouldn't have 5 or 6 kids that has 6 fingers. Like people are. Why do you say ew? I don't understand kids sometimes. This is natural. It happens. There's statistically somebody in this building was born with a tail, right? Like it happens. It's so. And obviously, as a child, as a baby, you would have it removed and whatever. But that's not the point. Is that these mutations do exist in the population. So you'd expect to see them. Some of you, those of you that do decide to continue on with your education, become physicians, you will see this in the hospital. Babies born with 6 fingers or babies born with tails or whatever.
Especially me.
All right.
When you say a tail, like,
No,
what do
you
you mean by
said you gonna
tail?
see that.
Like a tail. And we'll talk about that later.
Like, how you did with its whole tail.
Is it like a lion tail?
Like, it's
In different lengths. Some of them can move. Some have muscle attached. Some don't move really. They get removed quickly.
Oh my god. Like you probably make back like
Not like a. All right, can we not
I'm
focus
thinking
on
like
that?
a lion. Like, exactly.
It's a little. Usually they're about this tall. I can show you on.
oh, so the
you
tail's
can look
gonna
it
be
up.
like extra long?
I think like a lion tail.
Why don't I let you guys do that on your own during lunch? You can look up people with tails. You can look them up. They have them. The only question. I'm going to warn you on the internet.
Mendoza's
There's
really
a.
speaking.
Yeah, please.
Mendoza's very student speaking. You may help you.
Oh, I hate that. It's the only thing with a beard.
Is you still stuff in there?
Okay.
No, you can spill. It pulls the water out. Yeah. What?
Regina gotta go home early, but she's not in here. She's in the
All right.
Oh
We'll
my
tell her when she gets back.
God.
Let me see.
Yeah. That's a Photoshop. That's what I'm warning you about. There are some.
You're like a dragon.
Oh my God! If you guys, can we focus, please? So tails, when you look it up, try to remember anything on the internet requires you to source it. To think about where the source is. Photoshop is easy to change things. There's a lot of fake pictures on everything. Just look at where the source is. I would look for medical pictures and medical websites and things like that. And you'll find some really good, clear, honest pictures of babies born with tails. And they are born with tails. People are born with tails. People are born with gill slits in their necks. People are born with holes in their hearts.
My grandma had that.
Yeah, so these things have to be changed. By doctors have to change this all the time. There are people that are born with both male and female parts, for instance.
Can you be born without any?
That's a good question. I don't know.
Any what?
Okay.
Any private part, genitals? Right?
Wait, stop speaking. Stop speaking. You're confusing. There's several different conversations going on at the same time. So everybody stop. So here's what's happening. It depends on what you mean. If you mean without testicles and ovaries, yes. Can you be born without a penis or a vagina? It's difficult to say because you would have to have some way to urinate. Does that make sense? So I'm not sure what the answer is on that. All right. So let's focus, because remember, we talk about genitals as if there's something about that they're just sexual. They're not just sexual. There's all multiple purposes, right? Rabina, you're going home, so get your things and go to the office. Yeah. Yes, please. Could you all just go one at a time, the restroom? But yeah. I know. I appreciate you asking, but there's obviously something going on where you all drank too much water this morning or whatever. That's fine. Just go to the bathroom one at a time. All right. Can we move on? So here you have this chromosome with all these different genes. If this is chromosome number 1, we number them. We number these chromosomes. If this is chromosome number 1, then in humans, let's say it's a human chromosome number 1 in humans, and this is where you find a gene that has to do with height, then you're always going to find that gene here. Is that clear? Wherever that is, on chromosome number 1. If you have the gene for insulin here on chromosome number 1, then in the same species, in the human, you're always going to find the gene for insulin here on chromosome number 1. Is that clear? Yes or no? This is extremely important for you to understand how things are inherited. You have to understand this. If you don't understand this, you're not going to understand the things that come after. Are we clear?
Yeah.
So, a chromosome carries a specific set of instructions that control a specific aspect of an organism. The gene controls something about the organism. How does it control it? It makes a protein. That protein does something. All right, we're all good with that idea?
Yeah.
So if this is chromosome number 1 from dad, then you also got a chromosome number 1 from mom because you have pairs, correct? Why do you say it that way? He always does that. It annoys me. So I'm going to call him out on it. So there you go. So you have the same genes in the same place. Why? Because their mom and dad were of the same species, right? Dad didn't come from Mars and mom from Venus, correct? Mom and dad were the same species. So you still have the height gene here. Pay attention, think, process, or you're gonna lose you're gonna lose where you are, and you're not gonna be able to answer the questions on the test. So skin color is also gonna be controlled here, and there's gonna be a gene here for toes. Is that clear? Now, length of toes, number of toes, whatever. Now, what's the difference between mom and dad's? I say this is mom's number 1, this is dad's number 1. What's the big deal? Dad could have A, T, G, C. Mom could have A, G, T, C. Right? Dot, 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 as it continues, right? Because genes are very long. You see how long they are. They're very long. I'm just using this abbreviation. Are we all clear on that? Are these the same? They're not the same. They're different. Correct? They are different. This is all in one person. This is you. This is your genome. These are your chromosomes. But chromosome number 1 from dad is different from chromosome number 1 from mom. Maybe you're tall. Maybe dad's tall and you're tall, and mom's short but you're tall. So maybe dad gave you this, and this happens to be dominant. So this is big A. Maybe mom gave you the recessive gene. She's short, it's recessive. Maybe she gave you little a. That's what big A and little a means. So when you did Punnett squares in middle school, this is what it was all about. Yeah.
So like, you know how like animals, like the lion and then
Yeah.
the mule or the donkey,
Mule, you're correct.
How would it work?
Okay, so we'll talk about that. Just give me a second. We'll talk about it. So, does everybody understand what big A and little a means? And that your DNA from your mom is slightly different. So actually, all of us have mutations. Talking about mutations, right? All of us have mutations. It's what makes us different from everybody else. We all are slightly different from everyone else. Your chromosome number 1 has the same genes as my chromosome number 1, as everyone else in this room. The difference is your chromosome number 1 is slightly different, has a slightly different code than mine does. And so, maybe not. Maybe we have exactly the same. Maybe we both have big A. But whatever differences we have in our DNA, they're slightly different. Various changes in a single nucleotide or several small nucleotides. These mutations make us different from everyone else on the planet. So mutations happen all the time. A lot of the mutations are what we call silent. They don't result in a change in the protein, so that you never see them. Some mutations give us an extra finger, or make us have diabetes because we can't produce insulin, or give us sickle cell, right? And help us be resistant to malaria, but also could give us some trouble down the road. So different mutations could give us different outcomes, but mutations we all have. Now, there was a question about ligers and mules, so pay attention because it's a good question. It is. A liger is one of those exceptions where a tiger and a lion normally do not meet in nature and have sex and have
They do
babies.
that in the zoo.
It might happen in nature, but it normally doesn't happen. They can happen in a zoo. Please, can we stop with the side conversation? It's not forced, it happens because they're both cats and whatever. So, can we just listen?
They gotta rebreed.
Wow.
Sorry.
So, here's the problem with that. Tigers and lions are different, and I don't know how many. I'm not going to look it up because I don't have time to look at this as a great example, so I'm not going to do this, right? But I'm going to go ahead and make up this situation. What if a tiger has 5 chromosomes? I know it has more, so. And on chromosome number 1, it has, the gene for a heart and the gene for lungs, right? And it has 4 others, right? And there's pairs, so it has 2 pairs of each, correct? Yes?
Yeah.
All right, so it has 5. Now, obviously, tigers have more, and I don't know what gene is on what chromosome. I don't care for the intent of explaining this process. Now, let's say a lion's a different species. There's no guarantee that they have 5 chromosomes. Maybe they have 7. Whatever, I don't care. Maybe their chromosome number 1 has a heart, but maybe their lung gene is on 4. Now, normally, they have 2 pairs, right? So this has 5 and this has 7, 2 of each. Normally what would happen is a tiger would meet up with a tiger, right? And it would give half its DNA to the baby, and the mom would give half its DNA to the baby, correct? Mm-hmm. So the baby would have 2 pairs of 5. That's what happens. You keep the number the same. We have 23 pairs. When you meet up with your husband or wife in the future, a long time from now, and you decide to have babies, if you decide to have babies, you will give 23 chromosomes, randomly organized, to your child. In women, you would put that in your egg. 23 chromosomes in your egg. The men put their 23 chromosome in their sperm. When the sperm and the egg come together, you have your 23 pairs, your 46. All humans have 46. That's what we do. That's how we continue to have the same species. Now, a lion, in this case, the baby tiger would have 5 pairs. The baby lion would have 7 pairs. But what if a lion and a tiger have a baby? Now, it can have any kind of combination. It can have 5. Maybe on number 7, it has the gene for brain. I don't know. I'm just making this stuff up. So their baby only has 6 instead of 5 or 7. They have enough to live. In fact, ligers are very large. They're oddly colored. They're kind of in between. The ones I've seen have been kind of, yeah, they're very large. They're very large, bigger than a tiger or a lion. However, the problem is they can't have babies. They can't have babies because they have an odd number of chromosomes. Now, when they try to have babies, their baby doesn't get the gene for brains, or doesn't get the gene for heart, or doesn't get the gene for lungs. Some essential gene is not transferred, so the baby is never born. So that's what happens with ligers, and in fact mules as well. Mules is a donkey and a horse having a baby. That happens with anytime 2 species have babies. It's possible they have a baby, but it's unlikely that that baby will have a baby. If they can't have babies, then they're not different species. Does that make sense? So that is the answer to your question. Yeah.
What type of lifespan do they have?
I'm not sure.
I'll look it up.
Can you read like 2, whatever they lifers?
I literally just answered that question.
I know.
They can't have babies. They're sterile. That's why tigers and lions are not the same species, because they can't have babies that have babies. One definition, there's many definitions of species.
Early 20s.
I wonder how long a tiger and a lion live. Yeah.
So you said the twins chromosome get like into an egg in the girl, so like when it's twins, you like that, like split or it's 2.
It's a wonderful question. I want to hold off on answering that question until we get to mitosis. Okay?
Tigers live 16 to 18 years, and lions live 10 to 14.
So they actually live longer, but they can't have babies. All right, so to answer your question, in the short term, what happens with twins is either the egg splits before conception and you get fraternal twins.
You got a minute.
So they share half the DNA, the mom's DNA. Or they split after the sperm and the egg meet and you get identical twins. But they split and develop into 2 different beings, either way. That's a short answer. We'll explain it after we go through mitosis. So, homework tonight, if you want to be prepared, nothing I'm going to grade. Homework tonight is you're reading, trying to get these answered. You're coming prepared for tomorrow. I'd like to do these activities this week. There's about half of this packet is activities. I'd like to get them done this week. Test next week. Well, we're going to do it in class, so you're ahead of the game. Turn in this packet before lunch is over if you want credit. And tell your classmates that they better turn in or they're not going to get credit.
What packet? Turn in what? What does it do?
I'll go with what she said. Yeah, go ahead. Sign out, sign in. All right. So guys, when we're dealing with genes, right, and we're dealing with these things, let's talk a little bit about mutations and how they happen, because yesterday someone asked about mutations. And this class is actually a little bit ahead. So we have a 5' and 3' end. And when we're transcribing, like I said, if you see this sequence of DNA, and it's A, T, G, C, A, T, G, C, A, T, G, C, A, T, G, C, right? It goes on. It goes on. And you see in the front, the very first page of your study guide, a small gene could have hundreds or 1,000 of these in a row. So they can really be very long. So the first thing you gotta do is, this is DNA. This is inside the nucleus, right? So let me go ahead and draw it inside a nucleus. So this is the nucleus out here. And the nucleus has pores, right, where something can leave, but not DNA, right? We're all good with that so far? So something can leave. What's the thing that can leave and not DNA? It's not DNA, because DNA can't leave. RNA is correct. So the nucleus is big compared to the molecules, but you imagine that these are 3 meter long molecules. These chromosomes are 3 meter. If you unwrap them, they would be 3 meters long, and you're fitting it inside a nucleus inside a cell. That's how much wrapping you do to get this thing compacted into a nucleus. So there's all these holes, all or we call them nuclear pores in the nucleus, that allow it to go from the nucleus to the endoplasmic reticulum where the ribosomes are. Correct? All right. So then here you have this gene. We have the thing. What's the thing that's going to come and read this gene? What's the enzyme that's going to come and read this gene? So this is DNA, and I should mention one more thing. Remember, this is a very long. I just got done saying it's very long. It's 3 meters long. So this is very long. It does have a 3' and 5', and it does. But this is very, 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 very long in both directions. This here is the gene. Right here is the gene. Gene 1. I don't care what it is, and it can keep going. It can be very long, but it's a gene. That's the gene. But there's parts of the DNA here that when I was in school, some people called it junk DNA. They called it junk. It was really not I don't want to say intelligent. It was not a thoughtful way of a thoughtful name, a thoughtful term. Because if you gave it any thought, you would know that life does not keep things around that have no purpose. If the DNA had no purpose, why is it there? Right? And that's what people asked. They said, wait a minute. We call it junk DNA because it doesn't code for anything. There's no information on there that's used by the cell. So it's junk. Well, it turns out it's not junk. In fact, one of my professors in medical school, he studied that DNA and the histones, the proteins that the DNA wraps around. And he said that people don't find, at the time, people didn't really find that there was a lot of use to study this part of the DNA. In fact, some people argued, why are we sequencing we sequence the whole human genome? Do you understand that? We know what every letter is on every chromosome in several different versions of the human genome. We know where all the different series are, the sequences are. Is that clear? We call it the human genome. We spend billions of dollars sequencing it. When I was a kid, it finished when I was in college. It took many years to sequence all that DNA. But people argued, why do we want to know? It's a waste of time and money. We don't want to know the sequence of DNA that this doesn't have genes on it. All we want to know is what's the sequence of DNA that have genes, right? Because genes are what make proteins. I mean, it made sense, right? Are you understanding what I'm saying? Because you guys kind of look like, there's at least one person's looking at me like they're confused, another person's kind of not, doesn't look like they're paying attention at all. And yeah, you look like you're confused. And your partner looks like she's like, oh, wow. Yeah. So, I mean, I'm looking at you, I'm talking to you, so I'm looking at you. So here's my question though. Why does it, it makes sense to me that somebody would say, look, this costs a lot of money. Why don't we just find the genes and just sequence the genes? Why are we worrying about this stuff that's in front of the genes and stuff that's after the genes? It doesn't really, it's junk DNA is what they call it. In fact, if you watch there's a movie that's on Netflix I was watching. It's a V-Wars, a Vampire Wars. It's such a stupid movie. Oh my God, I tried to watch it. I stopped watching it last night. I was like, I'm done. I watched 3. I held out one more than you. In the movie, they called it junk DNA, and I was just like, oh, why do you call it that? And they try to make them all look all fancy. They actually showed a molecule of DNA, like he's a really fancy doctor, so he can look at it. I'm like, I showed that. My kids can draw that. What are they doing? So, anyways, obviously they don't have a science teacher as a consultant because they have a lot better science props. But anyways, the point is they still, in the movie, they called it junk DNA. Probably the kid that wrote the script remembered that term from when he was in school, when they used to call this junk DNA. This is not junk. There's real reason to have this DNA, and there's a good reason to have it sequenced. It turned out there's a really good reason to have it sequenced, yeah.
Probably because it holds extra information that might be needed.
Excellent. It does. It does hold it. In a weird way, it does. It's not red. It's not that it is a gene. In some cases, they're old genes. They're no longer used. It's that this part here before the gene and the part here after the gene that's going to be before another gene. Does that make sense? If this part that's after this gene, down the road, downstream, we call it downstream, there's going to be another gene. And upstream over here, there's going to be another gene. But in between genes, you have these regions that the cell uses to guess what? Does anybody have an idea what they might use it for? Regulation, exactly. Regulation. It turns the gene on or off. You can turn this gene on or off based on this part. This part's the regulating gene part. This is where the enzyme, what's the enzyme called? RNA polymerase. That's going to be on the test next week, Wednesday. RNA polymerase is the enzyme that's going to land here in the regulatory area. There's all kinds of sequences, all kinds of biochemistries happening here in the DNA before the gene that makes the RNA polymerase land well, attach well, and do its job well or not. You can have little RNAs come in here and block the mRNA. You can have things that can block it. You have these things called transcription factors, right? They bind here. We call them transcription factors. And those are the things that bind in this area, and they encourage or discourage, if they're not transcription factors, but they block the mRNA polymerase or RNA polymerase. They can discourage or they can encourage the production of an mRNA and therefore the production of that protein. So you can turn insulin on or off, right? You can turn pigment on or off. Somebody asked about vitiligo yesterday, right? You can turn, I don't know if that's how vitiligo works, I don't know. But you can imagine you can turn the production of melanin on or off. You can turn anything, any gene, on or off, depending on what you're doing here in this regulatory area before the gene. So it turned out it was a good thing we transcribed everything because it let us understand how is it that you control the genes being turned on or off. So this is a transcript. This is the regulatory sequence upstream for the gene. Obviously downstream there's going to be a stop. It's going to tell it stop making mRNA. What a great question. There are similarities between these regulatory areas in different species. And so they do know that there's a similarity in sequence that you can say, oh, a gene's coming. But how do we know? We know the amino acid sequence, and because we know the amino acid sequence of the proteins, we can work backwards and we can find the DNA sequence. The short answer is we don't, right? But we know where some things are active and where some things are not, and we can figure it out using experiments. So we have to do a lot of experiments, a lot of careful study, and we can figure out where a gene is and what it does. All right, so it's not an easy answer. It's not like, we do have some fluorescent techniques that could Wherever there's active transcription, we can see the DNA glow, so we can literally narrow it down, and we can work backwards and find the sequence of DNA based on the RNA sequence, based on the protein sequence. So we'll look at that. I think we covered that a little bit with the universal code before the final last month. All right. So remember that once you make the mRNA, or once you make this RNA, then you have to take out the introns and put together the exons, right? Is that right? All right. So we have to come to. So the mRNA is always shorter than the gene. The mRNA is always shorter than the gene. I'll give you a clue. I'm going to ask that question on the test. The mRNA is always shorter than the gene because you're taking some of the sequence out. You're adding a little bit, this kind of thing called the poly-A tail. You add this poly-A tail, that's not really that, to me, that's not as important as you understand that this gene sequence, not all of it goes to making the protein. That's really important when it comes to mutations. You'll see why when we start talking about mutations next. But this gene is a gene. This is the part that's going to make the sequence, but not all of it is the message that gets to the protein. It gets to make a protein. Some of it is not necessary for a protein to make a protein. So that's it. What's happening in the nucleus, and that's important because I'm going to ask that question as well. Where's transcription happening? It's happening in the nucleus. That's where you're going to make. That's where the mRNA is going to be made. Then coming out of one of these nucleopores into the you see the tube, right? I hope it drew that well. It's more of a sheet. It's hard for me to draw, and I'm not that great an artist. I apologize. Okay, I'm not a good artist. It's not that I'm not great, it's I suck at it, but that's all right. But you have this sheet, this membrane that is the endoplasmic reticulum. It's attached to the nucleus. And on this endoplasmic reticulum are these ribosomes. So as the mRNA comes out of here, wiggles its way out of here, it's 3' end, it's 5' end, it's poly-A tail, it's shorter than the gene, the message is coming out. It finds the ribosomes and they attach outside. So where's translation happening? That's right. Translation happens outside in the cytoplasm, specifically endoplasmic reticulum, the rough endoplasmic reticulum, RER. All right, at the ribosome. That's all you need to know for transcription, and that's all the intro you need for translation. We're going to go on into translation next. But does everybody understand that part at least? All right. I'm going to foreshadow you that. I'm going to give you a little bit of foreshadowing. You can imagine if you change one nucleotide here, you're going to change the nucleotide over here. Does that make sense? There's really 3 big changes that can happen here that will make whole different set of changes over here. But before I can talk about the implications and the impact of mutations, we really have to understand what mRNA is and how you make a protein. We have to understand translation before I can talk about how mutations are going to... All a mutation is a mistake when you're copying the DNA. If I gave you a dictionary and I said, copy this dictionary, that's your job. I gave you a dictionary. And by the way, that used to be a job for people. We called them scribes. Right, you can't take a picture. If I told you to take a pen, then back in the day, that's how we made books. We didn't have printing presses. You had some monk or some educated individual who got paid with food, more likely than not, and prayer. They got a book. You sat there with a pen, a feather, a quill, ink. You dipped the quill in the ink, you read the book, and you drew it out here. That was they literally spent all day, and this is everywhere in the world, that's how people did it. The Mayans didn't even have writing with a pen and ink, they had knots. So they read the knots, And then they sat there and tied the right knots when they had to copy the book.
Wait, what do you mean when you say bend the knot?
Knots, like knots in a string. The Mayans had books, their written language was written in, you know how you have Braille where there are dots on a page? For blind people? Well, in the Mayans, they had their books, they wrote their, a really interesting and really amazing idea, is that when they wrote books, they wrote them in knots. So, space, it's the space in between the knots, and how many knots told you the letters and the symbols and the meaning and the numbers or whatever else was going on. They had whole books. When the conquistadores from Spain came to and invaded and killed the Mayans, they talked to the priests. They found out all their books were right there in front of them, but they couldn't even recognize them as books because they were just strings hanging on a stick. That was their book. That was their written language. Pretty cool, huh? So you got this mRNA, and let's review. You got this mRNA. It has a 5' end, has a 3' end, has a sequence of RNA, right? Not DNA. Oops, that's not right. And the sequence can be long, can be several hundred long. I'm obviously not going to draw several hundred nucleic acids here, correct? Understood? Is there you see anything wrong here? The T, good. I was wondering if somebody would catch it. So you have this mRNA. The message came from the gene. It was changed. Introns were taken out. Exons were spliced together. It came a poly A tail at the end. There's A at the end. A bunch of A's together in a row. All right, at the end. And then on one end you have 5'. On the other end you have a 3'. Everybody okay with that so far? So that comes out and finds a ribosome. There's two parts of ribosomes, a big and a small. I don't care that you know if it's what the size. There are places, there are actually positions here in the ribosome itself. There are actually little positions, little places in the molecule that fit the mRNA in 3 different positions. They get fit in 3 at a time. So this triplet, every 3, and I'm gonna highlight it in yellow, every 3 is a codon. So we're reading this 3 at a time. 3 nucleotides at a time. RNA nucleotides. So we put the very first one, AUG, and I think you were tested on this as well. We went over this and we tested on it. AUG is the start codon. I said you have to know this. You have to know how to read the universal code. I said you're going to have a universal code. And you have to be able to read it. You won't have to memorize the universal code, of course. You have the universal code and you're able to read it and find out what amino acids where. So let me paste it. So once we have, if we get rid of all this, right? We have a ribosome, so we know it's a ribosome. We know there's codons. We know the codons are on the mRNA. We know there's 2 parts of the ribosome, big and the small. It's a complex. There's a bunch of proteins, mRNAs or RNAs. These are the codons here on the mRNA, codons. There is an opposite matching pair. Don't we do that? Isn't that a theme we've been doing all along? DNA, ATG gets matched to TAC, right? We do that. We did it when we transcribed from GTA to CAU, right? We did that when we made the mRNA to begin with. We keep matching. They keep base pairing. We call it base pairing. We have 3. Each codon is going to have a base pair. What do you think we call that? The opposite of the codon. You learned the opposite when we said. We didn't call it parallel, we call it what? Because it's the opposite. We call it what? Anti-parallel. So this is not the codon, this is the opposite, so we call it what? Anticodon. Anticodon. The opposite of what you have. So if you have AUG, what are the three that you'd expect to see in this ribosome? Say again? I'm sorry, I can't hear you. UAC, right? UAC is correct. That's what you'd expect to see. Well, where is that UAC going to be? Because it's matching, and what is it? What do you think you need to? What are you trying to build again? Remind me. A protein. What is a protein? A bunch of amino acids put together in a specific order. What's the order? Well, there it is. This is the order. These are the codons that tell the ribosome what order. So this is the anticodon that's matching. But so far, all we're talking about are nucleotides. Where's the amino acid come in? Where is the amino acid? I'm going to show you. There's a bunch of these in your they're in the cytoplasm. We call them tRNAs. They're another type of tRNA. They're another type of RNA. That's right. It's a transport RNA. To me, it also looks like a T, but almost like a ghost T or whatever. Attached to this tRNA on this end is an amino acid. In this case, let's go ahead and use methionine. We have 3-letter codes for them. That's your methionine. On the, huh? That says what? MET. MET for methionine. Are we good? It's a lot, but I think it's not too hard. It's not conceptually hard. If you remember, the DNA's in the nucleus has to get out. The message has to get out. We have to change it to RNA. If you remember where you had a T, you're going to have a U. You're going to replace T with U. If you remember that A goes with T and G goes with C, or A goes with U and T goes with A, right? If you remember all that, and you remember that you got to cut out the introns, they get left out, and you're splicing them together. Remember that. Now you got an mRNA that goes into the ribosome that's in the cytoplasm. Where's translation happening? I'm asking you the questions I'm going to ask you next week. Where's translation? This is translation. Where's this happening? RER, where? Where in the cell? Is it in the nucleus? No, it's not. What's in the nucleus? DNA. Where are the ribosomes? In the cytoplasm that's in on the RER or free floating in the cytoplasm, but they're in the cytoplasm. We all agree on that? Ribosomes are made of RNA and proteins. They have with them 3 spots. The the start codon is known as AUG. These are the bullet points that you need to know for next week. So I'm telling you what you need to know. I'm telling you what I'm going to ask you. So what do you think, if this is carrying methionine, what do you think is on the other end of this tRNA? These tRNAs are in the cytoplasm, in the endoplasm. It's the anticodon. Just because we're running out of time, I'm going to answer for you. You have UAC on this end. So the ribosome, you remember that RNA polymerase, the video I showed you, a ribosome? I mean, of an enzyme that reads the DNA and makes that long mRNA really quickly? Ribosomes do the same thing. Say what? Huh?
Like this drawing?
Yeah, like that one, that drawing, that's correct. Well, this is going to happen, and this is the way it's going to happen. It's going to come in. The ribosome is going to match up, and look what happens. They match up. They base pair. Did you see that? They base pair. What does that mean? U matches with A with U, G with C. You see that? Then it clicks on over. There's 3 spots to this, so it clicks on over. And I'm going to go ahead and move it up a little closer, if you don't mind. Ooh, I don't know what just happened there. Let me connect all this. So what happens is that this ribosome takes, and you have these 3, UAC, that has this tRNA on it, right? This thing has methionine. Then the next one slips in. You have your mRNA here. And your next one's CAU. There's another tRNA. Each tRNA carries, each triplet code carries a specific amino acid. What is the next anticodon going to be? I can't hear you now. GUA, you say. GUA. Oh, I don't know why I put UUA. GUA. Why did you say that? Because C goes with G, A goes with U goes with A. Correct. Yeah? I would rather you not, but if you have to, go. So, GUA happens if you have a tRNA that has the anticodon GUA, then you have an amino acid called valine, VAL. And inside the ribosome, you bond them together. In the ribosome, you make a bond, a peptide bond between them. That's what this is all about, right there, creating the primary structure. Then the next one that comes in as it keeps ratcheting out, right? I'm going to go ahead and draw it again. And of course, there's a second one, right? There's just 2 subunits, so I don't want you to make a mistake. And so there you have your code. You have AUGs over here, and you got CAU, right? And then you have, what's the other one? GUA, right? No, it's not GUA. CAU, then, yeah, GUA. And then CUC. So now, here you got methionine, MET, connected to valine, right? Isn't that what we said? Yeah. Now we're connected to valine, VAL. Then you're going to connect it to a third one that's going to be what? What's the third one going to be? It's going to be CAU, correct? That's going to be connected to tRNA that has this, and it's badly drawn, but I'm going to go ahead, because for the sake of time, I'm going to go ahead and leave it alone. Otherwise, if I had time, I'd redraw it. So there it is. So what amino acid does, oh wait, did I draw this? Methionine, CAU. I messed up. The CAU is actually histine, so I made the classic mistake. And then GUA is valine. And then that gets bonded together. And before you know it, you have this long chain, and it could be very long because mRNA can be very long. This long chain of amino acid, connected 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 amino acid, et cetera, et cetera, et cetera, et cetera, right? Each different amino acid having different qualities, folding itself because of hydrogen bonding, so that you can get an enzyme that looks like this, another enzyme that looks like that, another enzyme that looks like this, right? So each shape, you can get collagen that forms skin, right? You can get keratin that forms nails. You can get digestive enzymes. You can get tubulin, microtubules. You can get intermediate tubule, right? Intermediate filaments. You can start building a cytoskeleton. Now you can start to make these proteins. Each protein doing a different function. How do you get different functions, different proteins? Because you have different messages in different parts of the DNA. They get turned on in different times, depending on the conditions that the cell is in when there's time to make them. Yeah, question?
I was asking, does it do the same thing for your hair?
Yeah, the same thing with hair. That's why That's how you can inherit curly hair or straight hair or wavy hair. You get the different hair textures, the different hair colors, all because you started with that DNA all the way back here in the nucleus. You started with the DNA, and that turned into an mRNA. They left the nucleus, they went to the ribosomes, that made it, that, by the way, got changed on the way out of the nucleus. Then it met up with the ribosome and made this long chain of peptide bonds that we call a protein. And if you have a lot of cysteines here, you got really curly hair. You don't have a lot of cysteines, you don't have a lot of curly hair. Or that's just a simple example, obviously. With enzymes, see, it's much more serious, right? If you have the right enzyme, then you're producing insulin and everything's good. You have the wrong one, you don't produce insulin correctly, and you have diabetes, type 1 diabetes, right? So disease comes from malfunctioning of either the gene or maybe the post-transcriptional modification, or something's wrong. It could be any one of these steps that could be wrong that could lead to a disease. Is everybody okay with what we just said? So there is a universal code. All right, so the question then becomes, how did I know that AUG was methionine? How did I know that GUA was valine? How did I know that? First of all, let's be clear, I did not memorize it. You have a periodic table in chemistry, you have a universal code. This is the code of all life on Earth. All life on Earth does this. Thank you for getting the door. I don't know who it is that's at the door. There's early college playing around? Unbelievable. So here we have what's called the universal code. This is, every creature uses this. This is what's amazing. And some more evidence about the thought of evolution, right? Think about it, the fact that bacteria use this, humans use this, trees use it, plants, algae. Every form of life on Earth uses this code, the same code. It kind of connects us all. So what do we find when we get to AUG? Who can find AUG? Let's A is what? AUG, let's take a look at it. Let's write it out. AUG. What's the first letter? A. And what's this? The first letter. So it's A. What's the second letter? U. And so A and U. So it's in this column, right? In this row, A and U is found in this box somewhere. And what's AUG? It's methionine, MET, also known as, let's be clear, start. Are we good? All right. Well, how do you know, how does the ribosome know that it's time to end? The end is not the last bit of the mRNA. The end is before the mRNA. When does the ribosome stop making a protein? Look on this chart and tell me when the ribosome stops making a protein. AUA. Wait, why AUA? AUA is here. That's isoleucine.
I said
Wait, what? UAA is stop, UAG is stop, and UGA is stop. Those are your three stop codons. This is where you stop, guys. This is where the ribosome knows how to stop. So let's be clear. I should be able to, on the test, give you, oh, by the way, this is a chart. You can make one for anticodons, but this is a chart based on the codons. Is that clear? What I mean to say is, if I give you DNA, this is what I'm going to do on the test. I'm telling you what I'm going to do on the test right now. You're going to have A, T, G, C, A, T, whatever, right? It'll be a different sequence. Then I'll ask you, what protein does this make? What protein is made with this code? What series of amino acids does this code make? And I'm going to ask you to ignore introns. I'm going to ask you to ignore introns, right? And I'm going to ask you just, look, here's this DNA. What amino acid sequence? I'm going to give you this chart. Obviously, you'll have this chart. Let's see if you can do it right now. Can you do it? Try it. Tell me, what's the series? Now, tell me. Do it. Do it on paper. Do it right now. Don't do it out loud. Do it right now. If you can do it. And then I'm going to do it together. I'm going to do it. And you see if we match. And if we don't match, you see where you made your mistake. Do it. Make an amino acid sequence. What's the first thing you got to do? This is DNA, right? How do I know it's DNA? It's labeled DNA.
That's what I was thinking.
And then what else? It has T's, right? So it's clearly DNA. That's in the nucleus. You need to get it out. We're going to ignore the intron step, the post-transcriptional modifications. We're going to go right to making the protein. So you go, first thing you have to do is what? What is the first thing you have to do if you have DNA? Turn to RNA. You have to do transcription. Correct. Excellent. I'm glad somebody's listening. So you have to do, T goes to what? A. Then A goes to what? U. Then oh, go ahead. There it is. Right? All right, so what's the series then of amino acids we have then? We have to find the start. And it's a 3 code, so we have to find the first AUG, which is what? The first one right there. Then what do? I have to read every 3, correct? So 3, the triplet code. 3, 3. So how many amino acids do I have here in this list? We're gonna assume it keeps going, but we have what we have, right? So how many amino acids do I have? 5. Okay. Well, what's the first one? What's the first amino acid according to this list? MET, methionine, correct. Is then connected to what? What's it peptide bonded to? What's UAC?
I can't either.
I can't either. Let me, hold on. I'm sorry.
It's TYR.
TYR, that's right. Tryptophan, right? TYR. It's okay. You don't have to know the names because you're going to read the chart, all right? Nobody's asking you to memorize names. Let's be clear on that. TYR. Then it's connected to what? The next 3 is UAC, right? Am I right? UAC was, I messed up the, I'm sorry, I messed this up. Next one's GUA. GUA is what?
Val. Like Valerie.
Oh, I hate this. My menu's old. So GUA is valine. Then, what's the next one? CGU, which is what? ARG. Do you see how easy this is?
Yeah.
It really is easy. It's going to be on your end of course exam. It's really easy to read. Yeah, there are some deep implications, but the stuff is easy. It's just matching it. Yeah.
So you're gonna give us one of those charts, you're gonna have to memorize that.
Oh my Lord! I
He
said
said he was giving us a chart.
Oh, I didn't.
I love you, but you don't. You guys,
My brain's
that
today.
I would never. First of all, I'm hurt that you would think that I would have you memorize a chart like this. Am I that awful that you all think that I would have you memorize this chart?
Yeah.
That just seems like, wow, you must think I'm a monster. All right. So anyways, there's 1, there's 2, there's 3, there's 4, there's 1, 2, 3, and 4. What's the last one here? AGA. AGA, AGA
It's hard.
is arg. It's arginine. That's correct. Look at that. You can repeat. Did you notice that? Did you? No, there's a couple other things you need to notice, but we can talk about that after break. The key is that you're able to do this. You're able to know what is the amino acid. Is it time to go? All right. Are we all satisfied that you can handle transcriptions? Is there anyone in this room that has a problem? Please speak up now or forever hold your peace, or at least for the weekend. Remember, all the work has to be turned in, any late work has to be turned in by the end of today. Remember, your final review, your semester final review is due Monday. You should have index cards made already. You had 2 days in class. God bless you and see you on Tuesday. If you have questions, see me Monday after your first final. If I'm here all day till 3:10. So if you have problems, see me on Tuesday during the day. Remember, after 12, you get to go home, but you can come in here and get tutoring if you absolutely need it, if you're confused about something. Have a great day. So again, I want to point this out clearly, that there is this redundancy in the genetic code. There is more than one tRNA that can code for the same amino acid. The implication is that you can have a mutation in the DNA and therefore a change in the mRNA that does not change the amino acid. You can change the DNA, the RNA can be different, the DNA can be different, and you end up with the same amino acid. That protects you. That protects you against mistakes. That protects you against environmental consequences, mutations that are bound to happen. But you can get a mutation that does not change your phenotype, that does not change what you look like or how you function. This is extremely important. This is extremely important. I want to say this again. We call these types of mutations that happen in the gene that don't lead to a change in protein. We call these silent mutations. So there is a change, but we don't see it. We don't. And of course, silent means hearing, but the point is it doesn't change anything else. Are we clear on this? Because it's going to be important. And tomorrow when we talk about mutations, and something's called frame shift, and there's others that are called point mutations. Those are the three you need to know: silent, point, and frame shift. And I think you'll see it if you understand this stuff. I think you'll see it clearly tomorrow. All right, so let's go ahead and look at this. Let's take a look at a silent mutation, and let's take a look at a point. All right, so let's take a look at a code, an mRNA code, or a DNA code. Let's go ahead and take a look at a DNA code. If you have in the DNA, so we're at the DNA level. By the way, tomorrow I'll have an activity for you to practice on. We're going to go through some of this stuff that I gave you in the study guide, and also have, might have to wait till the study guide stuff, but we'll give you the practice problems in class. So DNA is GCCA, but let me say it's A GCCA, right? ATG, not U, sorry, ATG. You know you can get repeats, right? Does everybody understand that?
Yes.
The genetic code is can have long repeats. You can have more than, they can put the same amino acid 3 or 4 times. Does that make sense? It's not about being unique at every nucleotide. Then TT. All right, so let's do these, let's convert these together. What, somebody tell me sorry, I saw 2 hands up, yeah. Go ahead. Hands up.
I was asking if the silent mutations, they're the ones that don't affect the phenotype, right?
Absolutely right. Because what is the phenotype? It's what you see. How do you see it? It has to be an amino acid change. Because DNA only makes proteins. It makes RNA. The phenotype could be an mRNA, because some of the mRNAs have different functions. We're going to get into that tomorrow. But some of the mRNAs make proteins, and if you change the protein, you certainly change the phenotype. So if you change the hemoglobin, you get sickle cell. If you change the melanin, you get lighter skin or darker skin, or no color at all in the case of albino, right? So if you change the protein, you've changed what you see as with your eyes because of that change in amino acid sequence. So somebody tell me the mRNA, because the first thing is transcription. So we're going from DNA to what? RNA. And we're gonna assume something. We're gonna assume there is no post-transcriptional modifications, right? No splicing in this section of the DNA. This is part of an intron. Is that okay with you? Because remember, the gene can be very long, but not the entire thing will be turned into an mRNA. Some of it will be cut out, and the rest will be spliced together. Remember that? So we're going to assume that this whole code is going to be part of the RNA. Is everybody all right with that?
Yeah.
All right, so let's go ahead and make the mRNA. What is the mature mRNA? What does it look like? CGG, good. UAC. UAC, UAC, and UAA. So what is CGG? What is it? Look at this chart. Can you see what CGG is? C. So I go to C. My first letter G. G. Arginine is correct, so it's arginine. Arg. What's UAC? Tyrosine, TYR. There's UAC in case you missed it, and CGG is here in case you missed it. There's arginine, there's tyrosine. What's UAC? Tyrosine, so you got 2 tyrosines in a row. What's the next one? Tyrosine, you got that in a row as well. So you got 3 tyrosines in a row. Then what do you get? Then you get a stop. Then it stops. That's it. You know, this could be a very long protein, but the very end of it is arginine, tyrosine, tyrosine, tyrosine. Is everybody okay with that?
Yeah.
All right. So what if we have a mutation? What if we have a mutation right here? Let's see. What if you have a mutation here? Can you see that? Is that dark enough for you to see? Mutation right here at this G. What if this G in the DNA, some UV light, some radiation, some cigarette smoke, something changes this G from a G, right? No, not to an A, from a G to a T. So you have ATT. What do you get? You'll get a stop, is right. But let's go ahead and do it. The new mutated version, and I'll write it in yellow, the new mutated version would be CGG again. That doesn't change. UAC, right? UAC. But now the last one is not UAC. It's what? Last was not. It's not UAC. What is it? It's UAC, UAC, but then what? UAA. And the last one's UAA. But yeah, you stopped it early. You truncated the protein. You made the protein shorter. So now you made that protein shorter. Now instead of four amino acids, instead of arginine, tyrosine, and then tyrosine again, you have stop. You just changed that protein. What does that mean? I don't know. It could mean that you were dead. You were never born, right? It could mean that you're short. It could mean that you're really tall. It could mean that you have big eyes or small eyes. It depends on what this protein does. What does this protein do? I don't know. Yeah, question? Say that again? That means it stopped. It's not making protein anymore. It tells the ribosome, stop making protein. We're at the end. We're done. And so I just took off one amino acid, but some of these mutations you could take rid of most of the protein. If you have a mutation at the beginning, if you had this UAC, think about it, if this G turned into a T here, then it'd only be one amino acid. Like you'd get rid of 3 in a row. Yeah. I can't hear him because you're talking. You're literally sitting next to me. Say that again.
So each amino acid, like seriously, they all have they do different stuff.
Yeah, every amino acid is going to change the property of that protein. So in this case, this kind of mutation is a point mutation. Specifically, you need to know that, so you might as well write it down and learn it. This is called a point mutation. That's when you have a mutation in one amino acid. It just changed, one nucleotide. A point mutation is when you mutate just one nucleic acid.
So like all those amino acids come together in the gene.
Or I'm sorry, not one amino acid, one nucleotide. Go ahead, sorry.
So when each amino acid come together to make a gene, when people do gene changes or whatever, do they like read what all the amino acids do? Do they like try to change it?
Well, I'm going to talk about that in a second. Point mutations can give you all kinds of different outcomes, right? There's 2 possibilities from this point mutation. You can get a missense, which means what? What do you think missense means? All right, so sense means making, what does sense mean? Sense. You make no sense. Right, something that has meaning, right? Sense means meaning. What does miss mean? Not there. So this missense means there's like a misunderstanding, right? So you have a mismeaning. You have a misunderstanding. You have a mismeaning. So in missense mutations, when you have a point mutation that changes the amino acid. So if you can have a silent mutation that we just talked about earlier, right? So 1, it could be a missense. 2, it could be silent. You can change make the mutation, but it doesn't change the amino acid. Or if it does, we call it a missense, right? If it's silent, you don't see any change. If it's a missense, you've changed the meaning, and the meaning doesn't make as much sense anymore. You've changed the amino acid. Does that make sense so far? All right. Hold on a second. Let me say one more thing here. The other is you can have a nonsense. That's what I have to deal with every day in school. Nonsense. I deal with nonsense all the time. Nonsense mutations means you get a stop when you shouldn't. An early stop. So missense is when you change amino acid. Silent is when you change the nucleotide, but there is no change in amino acid. And a nonsense is when you have a stop where you shouldn't have a stop. You make the protein shorter. Yeah.
How would you know how long it should be?
You don't. It's whatever works. There's no should. It's whether it works or not, right? Mutations are, we can look at the human species and we can say everybody's DNA, everybody's gene on this, everybody has this, that is healthy, has this gene sequence. When you see this amino acid substituted, now you have sickle cell, your hemoglobin's bent and you got sickle cell trait. If you have two versions of this gene, you have sickle cell disease. So what you see is, again, here what we're talking about is what we see is happening, and it's a great question, like how do you know it's what you're supposed to have, right? And what we say is what does the average person have, and how does it work for them, right? Like how do we say that this person has a mutation, this one doesn't? They both have mutations. The question is which one has which one's part of the larger population, and which one is a part of the lesser bit of the population. Is that making sense to you? Yeah. All right. It's hard to explain. Yeah.
So it would be 100% of our baby will have a disease if I like had kids and I have traits.
Let's talk about that in next week when we're getting into the genetics part of this. So this is all genetics, but this is the foundation of what we're going to cover next week, which is the different kinds of how do you inherit this stuff? And that's the question you're asking. So it's a great question, but let's hold off to tomorrow if you don't mind, Ashley. Oh, sorry, didn't mean to use your name. All right, so that's when you change the DNA, when the DNA is changed in this way. But there's another way that can happen, another thing that can happen in DNA. Here I'm talking about what? I'm talking about changing this nucleotide, right? What else could I do to this sequence? What could I do to this sequence? What else could I do to this sequence? I just changed this G to a T. What else might I do? What could I do to it? What mistake could happen? Yeah. I can't hear you. Okay, so this could be this start here, this AUG somewhere over here. Could it's not here, but it would be, so it could be further up or later. Okay, you can change the start. That would be a missense again, but that missense would change the reading frame, which we'll talk about in a minute. The question though is, here's this DNA. I've talked to you about changing from G to a T, or G to any one nucleotide to another nucleotide. I'm not changing anything else. I'm just changing that one nucleotide. What else could happen to the DNA? Think about it. The answer is obvious, or I wouldn't be asking you. Say what? So you have this DNA sequence. It is what it is. That's the average DNA sequence. Let me go ahead and use names. Carly, here's this DNA sequence. Here I've been talking about changing this A to a T. What else could I do to this DNA to change the sequence? Instead of changing, what could I do? This is replacing it, so that's good. So replacing is one thing I could do. What else might happen? I can't hear you. Okay, I could change something up here and turn this into a stop, and that could stop, and it never happened. That's true. But what else? I can cut it. You break it, right? You can delete it, right? You can delete it. Something could happen. The DNA could break, and the cell gets rid of that DNA. So you can lose it all completely. We call that. Can you guess what we call that? Make a guess. Give me a name. A deletion. You can get a deletion. You can get a whole piece of the DNA just cut out and never put back together again. It can happen during splicing, right? When you're cutting out introns and exons. Or it could happen because of the chromosome itself could break off. We think that, it's a long story, but if you look at ape DNA, gorilla DNA, and human DNA, they have extra chromosomes, but you can see where their genes, the extra chromosome they have, you can see where it broke off our chromosome. So you can look at our chromosomes, you can actually map the genes that go there, and the sequences are very similar. You can see that where they have two, where we have one chromosome, they have two chromosomes, two smaller ones, and they match up. So that was a break that turned into a whole chromosome, that turned into a new species.
Do you think it's have the technology to do that themselves?
Yeah, we can do it. It's not something we want to play with, but yeah, we can do it. Is that, we call it CRISPR. CRISPR is a DNA, we can do anything we want with CRISPR. Huh?
When that lady came.
Yeah, when the lady came here, she talked to you about CRISPR, yeah. All right, deletion. And actually, Carly actually actually saw some CRISPR process going on in the lab that she worked on past summer. She didn't know it at the time, but didn't understand it. But CRISPR is just a biomolecular technique that we use, that stuff that you learned about in PBS or whatever it's called, where you learn how to use enzymes in bacteria to cut up DNA. And CRISPR is a mechanism that's very specific. It lets you change the DNA any way you want. You can replace. There's all kinds of techniques, but CRISPR is one of the most efficient that we have. So deletion is another kind where you can just get rid of. What else could you do? You can delete something. You can change it. You can make it nonsense. It can be silent. The change could change the amino acid. What else might you be able to do besides deletion? Add something. Excellent. What do you think we call it?
Addition.
Addition would be a good one, but no, it's not called that. It's called this. I mean, it's very similar to addition. They could have used addition. All right. And then there's one more, and then we'll have to talk a little bit about some generalities. Obviously, we're going to have to move things around because this mutation discussion is taking longer. So insertion, deletion, nonsense, silent, missense. There's another kind of change in the DNA. Yeah, I see your hand up. Silent. I kind of want to wait on going back, but insertion, there's a kind of insertion kind of similar to insertion. Instead of inserting where you insert in the middle, you could do something else. What might you be able to do? Yeah,
Maybe you could add to the vertical.
yeah, you could add and you can copy. It can be copied. You can miscopy and make, and instead of just adding the amount of DNA that you should have, you two are going to lose your phones. You're going to be very upset. I don't know what you're doing back there. I don't know what sneaky little business is going on, but it needs to stop. So you can take this and you keep looking at it like, what am I supposed to do with that? I don't know what you're doing. I don't know what you're doing. I don't care if you're playing tic-tac-toe. I don't care if you're texting. I don't care. You're not focused on the science. That's what I care about. So you can add on to the chromosome. You can add on to the DNA. And we call that duplication. So that's another way, another change. So those are 6 major ways. Obviously, there's all kinds of little things, little subheadings, and little sub things you can learn over time. But there is a type of mutation. It especially happens when you get an insertion or a deletion. Especially happens in insertion, and also obviously with duplication. But when these 3 things happen, something happens that really radically changes the DNA. And hopefully I'll be able to explain to you how that is. All right. Are we done? Can I go on? Yes?
Yeah.
I don't see anybody saying, no, please, I don't understand something. All right. Oh, did I just... All right. So here again, you have this amino acid sequence, right? And how do we read this? We read this with a reading frame. A reading frame looks like this. It's whenever, has anybody ever done any classic photography, any development of material. Have you ever seen like an optical little magnifying glass that you look at? You can look right on a light table. You ever done any of that? Yeah, that's the problem. I'm trying to think of how you might be able to, what you might do in your life. I know what I've done in my life that's very similar to this, but I don't think you have many things that you do. But if you can imagine that you have a frame, if you do this with your square, and you have a little frame, this is how you're going to read. Do you have older people like myself in your family that need to read with magnifying glasses? Or little special reading glasses or something? You ever seen those little? All right, I'll tell Miss Simpson you said that. So you have these little. So now you have the you have those. They have those little loops that old people use like myself to read. They have like their magnifying glass. They look like little squares. They read it. There could be all this tiny little writing here for there as far as their eyes are concerned. The writing's real tiny, they can't read it. But then here they see A, G, T, U, right? So this is their reading frame. Reading frame. In other words, this is what they see. This is what they can read. Does that make sense to you? Yes or no? All right. So this is what they read. This is all the reading there is. This is all the information there is. But the only thing they can read is what's inside this box. And they read it one at a time. So they read this, then they move it down this way, and they read that, right? And they move it down some more, and they read that. Do you understand what I'm saying to you? That's how we old people read when we're reading things that are very small. Is that fair to you? Do you understand this? Because if you don't understand this, you're not going to be able to make connections. You don't make connections, you're not learning. All right. So that's a reading frame. Well, what's the reading frame that a ribosome uses? What reading frame is the ribosome going to use here? It's reading what? Yeah. Almost. It is reading 2 at a time, but it's not 2 at a time. It's what? 3 at a time. It's reading like this. 3, then 3, then 3. You're right, it's reading pairs, right? It's matching pairs, but it's reading 3 at a time. Does everybody see that? Okay, so that's the reading frame. But what happens when you insert something? So let's look at, like when I change something, if I change, like let's say I'm changing UAC. UAC is tyrosine, right? If I take this C and I change it to a G, no, instead of UAC, it's UAG. Now all of a sudden I stop, and that is a nonsense mutation, correct? But I'm just changing one. If I change UAC to UA, unless that's not a good, GUA, so GUA is valine. If I change it to GCA, if I make 2 mutations, take GUA and change it to GCA, I just changed this amino acid to alanine, right? That's a missense mutation. But look at what I did. What I'm talking about here is a change in one amino acid, just one, right? I changed 2 nucleotides. I go from valine to alanine. That could make a big difference in the function of the protein. It might make the protein not work at all. It could make the protein work better. It might not impact it at all. You may not. So if this were melanin, let's say, you might get an albino person, you might get a darker person, or you might not get any color change at all. You might not see any change in color at all in the person, because it all depends on what this one amino acid does. Is that clear? But now look at what happens if you have an insertion. What if instead of changing these 2, I just add 2? Let's go ahead and do that. So AUG, UAC. Now instead of changing, I'm going to add AA. and GUACGUAGAUA. Everything else is the same. I just added 2. What just happened? AUG is the same. UAC is the same. Now I get AAG. That changed that amino acid, didn't it? It used to be GUA. Now it's AAG. But guess what? You shifted the whole reading frame. You didn't just change one amino acid. Because look at what's next. UAC. It used to be CGU, now it's UAC. GUA. Used to be AGA. GUA. Used to be UA something. Do you see what just happened? You inserted something. You inserted 2. If you insert 1 or 2 nucleotides, just 1 or 2. If you insert an even number, less than 3. If you have something that's not a multiple of 3. If you insert 3, then the reading frame stays the same. If you inserted 3 here instead of AA, you inserted AAA, then GUA would have stayed the same. Because remember, it's reading what? Every 3. So if you insert something that's not a multiple of 3, you're going to change what's called the reading frame, and we call this a frame shift mutation. Frame shift mutation. Yeah.
Would the ribosome be able to read it?
Yeah, it reads it. It reads AUG, no problem. Then instead of UAC, instead of getting tyrosine, well, it gets tyrosine because that stays the same. But instead of getting valine, what would you get? This is still methionine. This is still tyrosine. This AAG is what? Look on this chart and tell me what AAG is. Keep talking back there. See what happens.
Lysine.
Whatever it is.
I can't.
LYS. It's
LYS.
LYS is lysine, so LYS. Then UAC is what? Tyrosine. You don't have to know these, obviously. Tyrosine. AAG, AGA is what? Arginine, ARG, and then whatever. So now, do you see what just happened? You used to have methionine, tyrosine, valine, arginine, arginine. Now you got methionine, tyrosine, lysine, tyrosine, tyrosine, arginine. Now you've changed the protein significantly. So frame shift mutations change the protein significantly. That's the take home here. Where a point mutation might change one amino acid because you're shifting, you're changing what. And yeah, you can get a nonsense that truncates the protein, makes it shorter, right? You can get a mutation, and I see some people, oh, still the same behavior, huh? So sorry. Even after all I discussed with you. Oh well, life will teach you the hard way. So a frame shift mutation. The frame shift mutation is a shift in the entire, it can change the entire protein. With this kind of mutation, you're more likely to get a significant difference, a disease, or a significant advantage, because mutations don't necessarily lead to a disadvantage. They could lead to an advantage, right? Mutation, just like the X-Men, right? Mutations don't have to be bad. Although I don't like the X-Men because it's ridiculous. There is a good lesson there. Yeah, question? Yeah, very good, thank you. When you come in tomorrow, you will have an activity. Be ready to do the activity. You'll have the whole period to work with these ideas. I hope you studied and you paid attention. I will post this video again tonight. The entire video will be posted. Yeah, question? Huh? You can't quickly. I would just write this down and just share right now, for right now. So, the very first page, page 88, there's a cover, obviously. There's the cover. The cover is not meaningless. The cover has key terms. You should, if you're doing the right thing, you should be highlighting words that you know the answer to, that we've already discussed. For instance, I would have already highlighted amino acids, anticodon, base pairing, coding strand, maybe not, codon for sure, DNA, exons, introns, nucleotides, nucleic acids, protein, RNA, mRNA, RNA, tRNA, start codon, stop codon, transcription, translation. Those words I would have already highlighted. Notice I skipped all the DNA replication stuff because we have not covered yet DNA replication. The copying of the DNA we have not talked about yet. We'll cover that next week. So obviously, do I expect you to have that done? No, I do not. The other thing I've not covered yet is the DNA technology. The DNA technology, is it showing up?
Oh,
on the
the lights need to get
All
on.
right, turn the lights up. So those have not been covered yet. So obviously, there's two big topics we have to cover still. But still, you would know that if you read the cover. You have a bunch of check boxes there. Check them off if you're doing them. If you're not doing these check boxes, then you got problems. Can you describe and explain the main functions of the genetic code? You should be able to do that by now.
Yeah.
Can you identify 2 stages of gene expression? What are the 2 stages of gene expression? Transcription and translation. Can you identify them and explain them? At this point, you should be able to. So if you can't, then that means what? You got work to do this weekend. If you can, you got no homework to do, right? So you're in a good place. But how much work you have to do depends on how much preparation you're doing. You have to be reflective. Recall the structure of amino acids and how they form primary structure proteins. Describe and explain translation, including the roles of tRNAs, ribosomes, start codons and stop codons. So we'll go on. But those are the kinds of things you should be able to say, yeah, I know that, or I kind of do, but I don't really. I need to work on that. First page, page 88, we answered all those questions. They should all be answered, done. If you don't have that answered, you need to work on that. We've used it as an example in these notes. We've talked about it. It should be complete. Page 89 should also be done. Repeatedly, I've answered questions off that page in class. I've given you the answers. Describe 2 important ways in which a prokaryote and eukaryote's chromosomes differ. I've told you that. So I'm not going to go through it again. And if you don't remember, where can you find the answer?
On YouTube.
On YouTube or where? Where is the easier way? Right in the paragraph above it. The answers are written on the page. Page 90. Plasmid DNA. Talked about in class. Discussed it. Did I discuss all the detail of plasmid DNA? I discussed what a plasmid is. I explained how a plasmid can convey an advantage. What advantage? Antibiotic resistance in bacteria. I specifically go review this video if you don't believe me. Why are plasmids such as TI plasmid useful to genetic engineers? We haven't covered that yet because we have not covered DNA technology yet, and we'll discuss that at that time. And that goes with the second part of that question as well. But also the answers are there if you need to. Is that clear? So I'm going to go ahead and put two-thirds of a check on this one. 2/3 of page 90 should be done, if not the whole thing. 91, you should have read about it already. Something called chromatin's DNA chromosome structure. I told you DNA is wound up 3 meters long, wound up in a nucleus, wound around proteins called histones. Repeatedly mention that. And they're there. They explain there everything that's going on. And they give you really great diagrams. You should have read it by now. There's nothing to do on that page except read and take notes. You've had this for a couple months now. We're only on page 91. You should have done that. Page 92. Explain the significance of terms that used to describe the structure of chromosomes. What is DNA? What is DNA? You should be able to describe. Chromatin is just unwound DNA. And we'll talk more about that after we're done with translation. Histones, centromere, chromatid. We will talk about that later when we're doing this DNA replication business. Explain human cell. Each human cell has about 1 meter length DNA in its nucleus. Discuss the mechanisms by which DNA is packaged in the nucleus and organized in such a way as not to get ripped apart during cell division. Again, we have not talked about DNA replication or mitosis yet, or meiosis, so those are things that are going to have to be left for after the test. Are we clear?
Yeah.
So, page 92, you have about half that work should be done. Page 93 should be completely done. So what is a karyotype? We talked about chromosomes and how chromosomes, having a specific number of chromosomes are very important, that each chromosome has a specific set of genes, and those genes, you have to have those genes in order to be successful as an organism, and that you have 23 pairs, one copy from each parent, and there they are listed. Do you see them there on page 93? Do you see how they have a specific banding pattern? Do you see how they have a specific size? And they're all numbered, and there's 2 of them. We'll talk more about these chromosomes in a minute. How many chromosomes does each organism have? They're all listed there. Do you think you need to memorize how many chromosomes each organism has? No, you do not. But you should look at it. You should notice some things. Yesterday, a student said, "Hey, wait a minute. How come humans have so few chromosomes? Look at a dog. How many chromosomes does a dog have? 78. If DNA is so important, why does a dog have 78 chromosomes and a human only have 23 pairs? We only have 46 chromosomes and a dog has 78. If DNA is the end-all be-all, how is it that dogs have more of it than we do? And we're more complicated, we're smarter, blah, blah, blah. Corn has 108 chromosomes. Corn doesn't even have a brain. Yeah, is it smart? Does that mean corn's smarter than us? It doesn't. The answer is in which genes are turned on, which are turned off, and how do you manipulate those proteins? How well do you modify the proteins after they're made? How well do you modify the mRNA? How do you have introns and exons? How do you change the signal? Because the signal is not just the message in the DNA, it's also everything that comes after it and how you adjust it, how you express it. It's how you express the proteins that matters. It's not how much of the DNA you have. So be careful with that. But you could have thought of that on your own had you read this. And there's some questions there. What is karyotype? What information can it give you? What are autosomes? And what's a sex chromosome? So again, you're expected to have that answered. So page 93, I expect all of that done by now. 94. It shows you how scientists get the chromosomes, how they identify which chromosomes which. And it's just an activity that you can do on your own. And I expect that done. You can do it. If you have any questions, you can ask me on Monday. Question?
That's the best one.
Yeah. I think someone else had to go first. Did they go already? All right.
Number Kylo.
Page 95. Page 95 has a bunch of chromosomes underneath the microscope. Right? That's what this is supposed to represent. What are you supposed to do with these chromosomes? Cut them out and do what with them? Organize them by size and banding pattern. But how are you going to identify them using banding? Where do you see the answers for their banding pattern? Where are you going to look? Page 93, the previous pages. Page 93 gives you the banding pattern. All you got to do is match them. This looks like a number 1. This looks like another number 1. This looks like a number 2. Remember, all these chromosomes you see, each have a different set of genes, and those genes are very important. I wanted you to do it in class today, but obviously we're not gonna have time to do it in class today. So just do it for homework. I think you can handle cutting those out and gluing them and or taping them, correct? So if you have, anybody have problems with that? Okay. And if you don't have glue, you don't have tape, then staple them. You don't have staplers, tapes, or whatever, then just cut them out and put them each in their own little envelope. 2 little number 1s, 2 little number 2s, 2 little number 3s. You get 23 envelopes, 23 little pockets or something. You can separate them, number the back of the chromosome. Figure out a way to get the work done, and when you come in on Monday, you can glue it or staple it here in class. You don't just not do it just because you can't get it done perfectly. You do it anyways. So that's page 97, 95, 96, and 97 should be done by Monday as well, correct? Does anybody see a problem with that? This should just take you a little time. You can even do that in front of the TV, for God's sakes. It's like a jigsaw puzzle, right? So that's not really that difficult. Page 98. This again is just a matter of reading the stuff that we've already covered. We already talked about this. But this is talking about the experiments that let us know let us know that DNA is carrying the information, the genetic information. So make sure you read it. Make sure you read it. I expect it read, notes taken, and ready for discussion on Monday. 99, same thing. Read it, answer those questions. Page 100, RNA molecules. Now there's some details there I want to discuss in class. You can go over it. You can try to answer them yourselves. I know at least 4 kids have submitted already their extra credit. They've already done this. I'm okay with that if you've done it. But honestly, page 100, I kind of want us to do it together. But if you've done it already, great. You can read it. It's not that difficult. But 101, again, it's an activity. I think those 2 activities, the chromosomes and these nucleotides, I think those are 2 things you can do yourselves pretty easily. Guys, you're just cutting out the A's, the T's, the G's, and the C's, and you're putting the A here. What are you going to put next to the A? A next to C? A matches with C? T. All right. A matches with T, and G matches with C. Those are the base pairing rules. It's just that easy. I know you can handle it. Answer the question. It's just that easy. It's just not that complicated. You can handle that. So that takes us all the way to 105. 105 all the way through to, no, not to the end. 105 all the way through to 111. 105 through 111 we're doing next week. So don't do 105 to 111. We will do it in class. So this stuff here, this whole section should be done already, except for the stuff that I told you we're waiting on. This stuff you should get done this weekend, and it's just jigsaw puzzle-like activities that I think you can handle, maybe a few answers and questions. So, I don't know. To me, I think this is reasonable. Again, I wanted you to do the activities in class, so we ran out of time. Now, 112, 113, 114, and 115, 116, 117, 118, and 19, right? Pages 112 through 119. Pages 112 through 119. If you understand those pages, you are ready for the test. That's what your test is on Wednesday. It's on page 112 through 119. That's all the stuff we've been talking about. Transcription, translation, it's all there, step by step. Are we clear? So if you can answer that, if you want practice, these I'm gonna grade this as a practice test. And your test that's the answers to this section. If you've already done it for extra credit, well, you're gonna get double credit. Good job. But this is gonna be considered a practice test. The answers to these questions here, you get to do it at home. You get to use whatever resource you want. This will be due on Tuesday, not Monday. Your test will be on Thursday, not Wednesday, because we have this we lost today, right? Am I being fair? Does anybody have any questions or concerns about this? So this stuff you should have covered, except the stuff that I said we're gonna wait till after the test. This here is stuff that you should do this weekend, right? All the way, don't do this we're gonna do in class, so don't do this, DNA replication and all that. This is a practice test. This stuff we covered it, we took notes on, I went over it. You should be able to answer, and I'll even answer some of these on Monday even. If you didn't get them, you don't understand them, maybe you should ask questions on Monday, right? And then so Tuesday, these pages will be due, submitted as a practice test. You're either gonna get them right or wrong. I'll grade them as I get a chance. And then you'll have a multiple choice test on Thursday. That gives us Wednesday to go over stuff. And then we'll hit the rest of this, all right? Then we'll hit the DNA technology. We'll try to do some agarose gels. We'll try to run some gels, separate some molecules using gel electrophoresis. We'll talk about, I don't have any restriction on nucleases, so we're not gonna be able to cut up any DNA, but we can talk about how that works. Are we good? Does anybody have any concerns for the good of the order, or for concerns about academic information, about scheduling? Anybody have any kind of, I'm gonna be out of town this week, or whatever? No? All right. So I should not have any question. The practice test is from page 112 to 119. That will be uploaded by Tuesday. I will grade it. Thursday is your test, not Wednesday. Today was crazy and we got behind, so I'm moving the test back one day. Are we all clear on this?
Yes.
Then Friday we're gonna work on page 105 to 111, and we'll get on Friday we'll work on page 100, and by next week Tuesday you will not next week Tuesday, the following week Tuesday you'll have another test on DNA replication. So get that stuff done, get this work done. All right, you have this whole period to get this done. Let's go.